Good day, everyone. I am Ms. Marjorie D de Los Angeles, a faculty member of the Institute of Biological Sciences and a member of the DOST funded program, Assessment and Conservation of Forests over Limestone Ecosystem Biodiversity in selected municipalities of Samar Island, Philippines, also known as CONserve-KAIGANGAN. Before I proceed with my presentation, allow me to introduce the program and the members of Project One. The mission of the program, CONserve-KAIGANGAN, is to assess, conserve, and sustainably utilize the natural resources such as plants, fungi, bacteria, and animals in Kaigangan. Kaigangan is a Samar local dialect for forests over limestone. The program covers four municipalities of Samar Island, which are Paranas, Taft, Basay, and Giwan. The program is made up of four projects, and I am a member of Project One. Project 1 is Assessment of Biodiversity in Forests over Limestone. As reflected in my slide, here are the members of the project. Leading the program as well as Project 1 is Professor Inocencio E. Buot Jr. Ms. Elaine Loreen C. Villanueva is our Senior Science Research Specialist. Ms. Gina M. Mallison is our clerk. Assistant Professor Desmarie Antoinette P. Fernandez is our project staff. Mr. Paul John S. Tolentino and Ms. Ren Divien S. Obeña are our university research associates. There are 17 UN Sustainable Development Goals, and CONserve-KAIGANGAN as a research program covers some of these global goals. But without further ado, on behalf of the project, it is my honor and privilege to present a portion of our results entitled Ecology of Understory Plants of Forests over Limestone in Samar Island, Philippines. Once again, I am Ms. Marjorie D de Los Angeles, a project staff of CONserve-KAIGANGAN. Samar Island, together with Leyte, are collectively known as Eastern Visayas and is identified as one of the 15 biogeographic zones in the Philippines due to its unique and rich biodiversity. It hosts a number of native and endemic species due to its extreme rugged terrain, geographic location, climatic and edaphic conditions, biogeographic conditions, and a variety of habitats and forest formations, one of which is forests over limestone. Recent expeditions in these forests has led to new species records as well as the discovery and description of new floral species. Why study floral communities? A well-documented and conserved forest over limestone brings in numerous priceless ecosystem services that will sustain ecosystem health, minimizing global warming and drastic climatic changes, aggravating disaster in this vulnerable region of summer, as observed during typhoons. Furthermore, assessment of floral communities in the epigeic region of forests over limestone ecosystem can generate useful information such as floral diversity, community structure, niche partitioning, information on plant adaptations, as well as information on plant assemblage patterns. With these in mind, the following objectives were developed. Generally, the study was conducted to document and examine plant diversity and species composition in forests over limestone of Paranas, Taft, and Giwan of Summer Island. Specifically, the study aims to document plant understory species in forests over limestone and to describe patterns of plant distribution with reference to ecological parameters using multivariate tools. Assessment of floral diversity was conducted in Paranas, Taft, and Giwan of Summer Island. The study was conducted from October 1 to 16, 2019. The selection of sampling plots was based on the biophysical characteristics of the area, including plant diversity heterogeneity, topographic attribute, extent of forest over limestone area, and presence of anthropogenic disturbances. Materials and methodologies were chunked into five. Number one, processing of permits such as gratuitous permits. Number two, plant species assessment. Understory plant species were recorded in established plots. Pertinent data such as scientific name, local name, height, and cover of all plant species were recorded as well. Number three, soil analysis. Soil samples were collected and edaphic factors were analyzed such as percent organic matter, percent nitrogen, phosphorus, electrical conductivity, potassium, calcium, magnesium, iron, zinc, copper, manganese, and chlorine. Number 4, herbarium specimens. Herbarium specimens were prepared and filed at the Plant Biology Division Herbarium. And finally, data analysis. To detect significant changes in the plant floristic composition across select municipalities, data obtained from soil and plant were subjected to multivariate analysis. Results showed that a total of 26 understory plant species were recorded from the different established plots across the municipalities of Paranas, Taft, and Giwan. From the 26, 6 fern species belonging to 6 genera from 6 families were recorded, and a total of 21 flowering plant species belonging to 13 genera and 9 families were also recorded. Based from the forest floor census, highest species richness occurred in Paranas and was recorded to have a total of 18 understory plant species, followed by Taft with 12 and Giwan with 5. Paranas was computed to have the highest Shannon values and was deemed to be most diverse among the sites, and Giwan with the lowest Shannon values and was the least diverse among sites. A lower species diversity in Giwan may be attributed to the possibility that the forests over limestone in the area is still young. The analysis of importance values of species was computed for every site and summarized in tables 3, 4, and 5. Out of the 18 understory species in Paranas, the dominant plant species based from IV was Pandanus sp, belonging to the family Pandanaceae, followed by Cyclopeltis crenata, a fern species. Similarly, in the established transects at Taft, among the 12 species, Pandanus sp was computed to be the dominant species based on IV. The dominant plant species in Giwan based from IV is Tradescantia zebraina, belonging to the family Comelainaceae. Based from the non-metric multidimensional scaling, plant species composition were similar among plots established in the sites. Plant species composition were similar between Paranas and Taft, and between Taft and Giwan. These similarities can be accounted by proximity of sites. Environmental factors are equally important in biodiversity assessment and indicate overall environmental quality. Structural and functional characteristics of communities are shaped in accordance to habitat conditions. Interestingly, most of the analyzed edaphic factors are not typical of forests over limestones. Results from soil analysis revealed variations in soil chemical characteristics across sites. Soil pH values across sites were identified to be slightly acidic. Acidic nature of soils can be explained with the intensive leaching of basic cations brought about by high rainfall. Samar falls under type 2 and type 4 climate, where the former is characterized by having no dry season with pronounced maximum rain period throughout the year. Generally, phosphorus, percent nitrogen, and percent organic matter was high in all plots. Percent OM was highest in Giwan and lowest in Taft. Variation in average soil OM across sites can be explained by differences in vegetation cover, amount of leaf litter, and continuous rain promoting decomposition. The analyzed edaphic factors were found correlated based from the resulting canonical correspondence analysis, with the first two axes explaining 36.68% of the variation and patterns among understory plant species. Understory plant species recorded from Paranas and Taft are highly affected by iron and EC. Plant species from Giwan, on the other hand, is positively correlated with pH, percent OM, percent nitrogen, phosphorus, chlorine, potassium, copper, manganese, magnesium, sodium, and zinc. To summarize the results of the study, a total of 26 understory plant species were recorded across the municipalities of Paranas, Taft, and Giwan. From the 26, 6 fern species belonging to 6 genera from 6 families were recorded. whereas a total of 21 flowering plant species belonging to 13 genera and from 9 families were also recorded. Based from the forest floor census, highest species richness occurs in Paranas, with a total of 18 understory plant species, followed by Taft with 12 and Giwan with 5. A lower species diversity in Giwan may be attributed to the possibility that the forests are still young. Interestingly, most of the analyzed edaphic factors are not typical of forests over limestones. Soil pH values across sites were identified to be slightly acidic. Generally, percent %OM was high in all plots. Percent %OM was highest in Giwan and lowest in Taft. Variation in average soil OM across sites can be explained by differences in vegetation cover, amount of leaf litter, and continuous rain promoting decomposition. Understory plant species recorded from Paranas and Taft are highly affected by iron and EC. Plant species from Giwan, on the other hand, is positively correlated with pH, percent OM, percent nitrogen, phosphorus, chlorine, potassium, copper, manganese, magnesium, sodium, and zinc. Understanding key environmental factors are equally important in biodiversity assessment and can indicate overall environmental quality. On behalf of the project, I would like to acknowledge the following government agencies: University of the Philippines, Los Baños; Samar State University; Department of Science and Technology; Philippine Council for Agriculture, Aquatic and Natural Resources Research and Development; Samar Island Natural Park Protected Area Management Board; local government units of Paranas, Taft, and Basay; and Basaranan nga Organisasyon Han San Isidro Samar. Thank you very much, everyone. I am now ready to entertain your questions, comments, and suggestions.